Okay, I think we'll go ahead and get started. We've got just over 100 people online, and it's just 1 minute after 3 o'clock. So welcome to this overview of the Agriculture and Food Research Initiative, or AFRI, Animal Health and Production and Animal Products Program Area webinar. So my name is Deb Hammernick. I currently serve as the Director of the Division of Animal Systems, and I'm also the Deputy Director of the Institute of Food Production and Sustainability. So this webinar is being recorded. We will share a link to this, to the recording on the webpage where you found information on these AFRI webinars. We'll probably get that posted sometime next week. In case you need to go back and listen to anything else, or if you want to refer a colleague to that webinar that wasn't able to make it today, that information will be posted on the website. We'll also share the PowerPoint slides with everybody that registered to attend this webinar as well. They'll be sent to you in the form of a handout, again, probably next week or sometime soon after this webinar. So I want to remind you that this is a webinar format, so you will be able to type in questions in that Q&A box there at the bottom of your screen. If you type in your question, we have a couple of program specialists that will either type an answer to your question, or they'll read it aloud, and the question will then be answered verbally at the end of the webinar. But anyway, we would encourage you to type questions into that Q&A box so that we get them on record and we have an opportunity to provide answers to you. Okay, so next slide, please. So this webinar, as I mentioned, is going to cover AFRI programs in the Animal Health and Production and Animal Products Program area. And this is one of the 6 Farm Bill priority areas listed. So all of the base programs listed on this slide are relevant for animal science, animal health, and animal production. There are 6 programs that we consider the base programs. They're listed here. You'll learn more about these programs, as well as meet the national program leaders that lead and administer these programs during the rest of the afternoon. The 7th program listed there, Ecology and Evolution of Infectious Disease, is an interagency program that we participate in with NSF and NIH, and you'll also learn more about that program this afternoon as well. Next slide, please. Okay, then within the AFRI RFA, there are several cross-cutting programs that are relevant to animal production systems. And they're cross-cutting because they cut across the other 5 Farm Bill priority areas. So these programs will accept programs on animal science, as well as plant science, environmental science, food safety, nutrition and health, and so forth. But we certainly do want to make you aware of these programs because there are funding opportunities for animal science in these cross-cutting programs. And on the next slide, we'll start to go into an overview of individual programs.
Good afternoon, everyone. I'm Mark Mirando, and I work with the Animal Reproduction Program as a National Science Liaison based in Washington, D.C. So the program area priority code for this program is A1211, and we tell you that information because it's important to include that in your application when you apply to the AFRI programs. So that allows the application to get routed appropriately and correctly to the correct program. The other thing it's very useful for is after you've read the entire RFA and you need to go back to a specific program, you can use this program code as a very easy way to do an electronic search of the RFA. So the Animal Reproduction Program Area Priority request standard grants with a budget limit of up to $650,000 per project for 3 to 5 years of support. And this budget limit of $650,000 is for standard grants, strengthening standard grants, and new investigator standard grants. All the other grants I'll talk about have a different budget limit. And these budget limits that you'll see on this slide and for subsequent program are all the same. The budget limit applies to the standard grants, the strengthening standard, and the new investigator standard grants. The program accepts research projects only, and the grant types that are solicited in this program are standard grants, conference grants, and phase grants, which include strengthening standard grants, new investigator standard grants, strengthening conference grants, new investigator seed grants, and strengthening seed grants, equipment grants, and sabbatical grants. These are the only grant types. We do not accept coordinated agricultural projects or fellowship grants to this program. The application deadline is May 6th, so a little over 2 months from now, and the deadline is 5 p.m. Eastern time. And it's important to note that at NIFA, we adhere very tightly to the deadlines and the time deadline as well. So when the clock rolls over to 5:01 p.m., then your application is late and will not be accepted for review. My contact information, my email address is listed down there, mark.mirando@usda.gov. So the funding priorities for the animal reproduction program are listed on this slide. The program covers everything from cellular and molecular biology through genomic and genetic work to whole animal in vivo aspects of animal reproduction in order to improve reproductive efficiency or enhance reproductive management of livestock, poultry, and aquacultured food fish. The 4 specific priorities are focused on gonadal function, including production function and preservation of gametes, the hypothalamic pituitary axis, embryonic and fetal development, including interaction between the conceptus and the uterine environment, and the microbiome of the reproductive tract.
Good afternoon. I'm Steve Smith. I'm National Program Leader for Animal Production Systems, and I want to introduce you to our Animal Nutrition Growth and Lactation Program. It's structured very similar to the reproduction program that Mark just described, though the program area code for this program is A1231. Again, important to make sure that you apply to the correct program if this is what you're interested in. The proposed budget is the same at $650,000 for the total project, including indirect costs, for 3 to 5 years. We invite research projects only, and the grant types are standard, conference, and phase, with the $650,000 total, as Mark said, applying to the standard awards, whether they be standard, strengthening, or conventional standard. And then within phase, the strengthening standard, the new investigator, the strengthening, conference, and seed grants. Now, Mark mentioned it, but didn't draw a lot of attention to it. This is our first year for offering new investigator seed grants. So that's an opportunity where the difference being that under our old programs, you had to be from a strengthening eligible institution to apply for a seed grant. We're now opening that to new investigators that may not reside in a strengthening eligible institution. So that is unique this year. And then in addition to that, we offer the equipment and sabbatical grants. The application deadline is May 6th. And as Mark said, the 5 o'clock deadline is rigid. So I encourage you to please submit early. Very few people do, but start early planning your projects, set your timelines, and get them in early. You see my contact information there. If you'll go to the next slide, Mark. The funding priorities with the Nutrition, Growth, and Lactation Program are cellular, molecular, genetic, genomic, or whole animal aspects of nutrition, growth, and lactation, especially focusing on these 4 items: nutrient utilization and efficiency, including the influence and impact of the gastrointestinal microbiome; innovative approaches to feed formulation or use of novel alternative feedstuffs; improving the quality and efficiency of producing meat, milk, eggs, and animal fiber; and metabolic disorders and nutritional deficiencies affecting production of meat, milk, eggs, and animal fiber.
Thank you to Mark and Steve, my colleagues. My name is Frank Sievert, and I welcome you all to this session again. I'm going to introduce you to 2 programs here now. So the first one is the A1251, the Welfare and Well-Being of Agricultural Animals. And you're going to notice that this program here has a few added features compared to the previous ones. So first, when we talk about proposed budget requests, we're still talking about a 3 to 5 year timeline for these projects. $650,000, including indirect costs, is the standard, but there's an opportunity for partnership grants, which then we can add up to 150,000 more above the listed maximum. Now, if you go into the RFA, you're going to get a very clear description of what a partnership is, but essentially it includes significant collaboration with minority serving institutes, small institutions, small to mid-sized institution, EPSCoR state institution, and even international partners. So spend some time looking into that section of the RFA, and you're going to have all this information listed there. Also, in terms of project types, the previous 2 programs accepted research projects only. The Welfare and Well-Being of Agricultural Animals program accepts research and integrated projects. Now, by integrated project means that you have to have an additional focus other than research, and that could be extension or it could be teaching education. Again, the RFA will specify what exactly are the expectations for those. The types of grants are very similar to the previous ones. You have standard grants, conference grants, and then you have a number of phase grants in there: the strengthening standard, the new investigator, the strengthening conference, seed grants, equipment grants, and sabbatical grants. Those budget limits that are listed above, they refer, of course, to the standard grants, the strengthening standard, and the new investigator. The others have different values. Just like the previous 2 programs, the deadline is May 6th, the now famous 5 p.m. deadline. And both myself and Dr. Mark Mirando are the contacts for this program. So we're co-leading this program here this year, and both our email addresses are shown there. Next slide, please. So this program has a number of funding and priorities, and they all are built around evaluating current production practices or developing new management approaches to protect animal welfare and sustainable production efficiency. So you can focus your proposals on objective measures of animal welfare and well-being, including using emerging methods and metrics to assess outcome-based welfare. You also may approach your grant to look at alternatives or improvement for pain management, euthanasia and slaughter, handling, and transportation to decrease injury and stress. Also, we're interested in proposals that approach the understanding of the effect of the microbiome on animal welfare and well-being. You can approach it as a selection for robustness, improved behavior, or social effects. And also innovative alternatives to replace or reduce the use of animals in research. Next slide, please. Well, let's move to the second program that I'm going to introduce you today. And this one is going to take an extra slide because there's also a special feature in it. This is the animal breeding and functional annotation of genomes, and the priority area code is A1201. Now, the budget requests for these programs may either be $650,000 for 3 to 5 years on regular programs. But if you are submitting a project that addresses the USDA Animal Genome Blueprint, then that limit goes up to $1.3 million for 4 to 5 years. Now you can find that document online, and it's also going to be a link in the materials that we're going to make available. So I encourage all of prospective applicants to take a look into that blueprint to see if your project qualifies for that. On this program, we're going back to research projects only. So that's the only type of projects that we're going to be funding here. And the grant types are exactly the same as the previous ones that all my colleagues mentioned. You got the standard, you have conference, and all those categories inside phase grants. Again, application deadline is May 6th at 5 p.m. Eastern Time. And myself and Dr. Mark Mirando are the two contacts for this program for this year. So let's start with the $650,000 standard grants here. So what are we interested in seeing in the proposals? We want to see novel quantitative genetic methods that include selection theory, modeling, and implementation of selection methods. Also, the development of national and regional breeding strategies to address biotic and abiotic stresses, genetic diversity, germplasm storage and characterization, crossbreeding, or the modifications of the genome. And the development of new phenotypes for improving selection criteria and or development of high throughput methods for on-farm recording of data. Next slide. Now, this higher funding limit applies to the blueprint that I mentioned previously. And here the goals are a little different. So if your project falls into any of these 4 categories, we encourage you to approach this program here. So we're looking at genome-wide catalog of functional elements in both coding and non-coding regions of the genome, epigenomics, and chromatin architecture, among others. Genome design to use functional genetics or epigenetic variants underlying traits of an interest for traditional selection or as candidates for gene editing. We're also covering the comparative genomics approaches for genetic mapping of traits and understanding the evolution of traits to interest of commercial agriculture. And finally, the development of gene editing methods for high throughput functional screening of candidate loci in cells, tissues, or organoid systems. Thank you.
Good afternoon. I'm Kathy Bjork, and I'm one of the national program leaders for the Diseases of Agricultural Animals program. The program area priority code is A1221, that's A1221. And we have 2 lines of proposed budget on this, just as the others have, you've seen before. The $650,000 total per project for 3 to 5 years is for our standard grants. And then if you include a partnership in your proposal, you can request an additional $150,000 for those 3 to 5 years. I push you towards the RFA to look at the specifics on the partnerships. The project types are specifically only for research only. These are not integrated awards. These are research projects only. And the grant types, as we stated before, are standard, conference, and phase grants, including strengthening standard, new investigator, strengthening conference, seed, equipment, and sabbatical grants. As we've said before, the application deadline is May 6th of 2021 at 5 p.m. Eastern Time. And listed here are my contact information. I'm Kathy Bjork, and the contact information for Tim Sullivan, the other national program leader in animal health. Next slide, please. The overarching funding priority for this is the cellular, molecular, genomic/genetic or whole animal aspects of animal health and disease, with an emphasis on maintaining healthy agricultural animals. More specifically, we're looking for proposals on maintenance of homeostasis, including innate immune responses, disease prevention and control, including vaccines, reverse vaccinology, breeding for resistance, management and diagnostics. And note that this is for endemic diseases only. There are other programs for foreign animal diseases. Therapeutic interventions for disease, including minor use animal drugs. Development of publicly accessible, reasonably priced immunologic reagents for aquaculture with a major focus on catfish and salmonids or poultry, and the establishment of a research coordination network for minor use drugs. Some stats on our last year of proposals in FY20, we received over 140 applications for this program, and approximately 20 of these were from new investigators. For all proposals, the success rate was approximately 20%, and for new investigators, the success rate was a little higher at 24%.
Thank you, Kathy. I'm Tim Sol, National Program Leader in Aquaculture and Animal. And I'm going to be talking right now about the one interagency program that we have to discuss, which is the ecology and evolution of infectious disease. This one doesn't have a program area priority code, but it is an interagency program that has 6 partners: NIFA, the National Science Foundation, the National Institute of Health, UKRI, the United Kingdom Research Institute, and then the Binational Science Foundation in Israel, and the National Science Foundation of China. The proposed budget requests for this program are $2.5 million per project for a project that lasts for 3 to 5 years. The project accepts research project, the program accepts research projects only, and the grant types are standard grants. The application deadline is November 17th, and that's also a 5 p.m. deadline. I provided 3 contacts, so there are 2 folks at NSF that you can reach out to if you have specific program questions, Katarina and Sam. And then you can feel free to reach out to me if you have questions about NIFA's involvement in the program and what our overall priorities are. And we also provide the URL there to the RFA published on NSF's website. Next slide, please. So the funding priorities for the EEID program, it's really focused on ecological, evolutionary, and social drivers that influence transmission dynamics for infectious diseases. For these projects, there's a requirement that a central theme should be quantitative or computational understandings of pathogen transmission dynamics, and really a focus on zoonotic, environmentally borne, vector-borne, or enteric pathogens of terrestrial or aquatic systems and organisms. This includes diseases of animals and plants, and varies in scale from specific pathogens to broader environmental systems. Next slide, please. So Deb talked a little bit about cross-cutting programs, and I'm going to be introducing the first of those, which is the Tactical Sciences for Agricultural Biosecurity program. And then other folks will talk about the next. So the Tactical Sciences program has a program area priority code of A1181, 1181. For the proposed budget requests, projects that have a single function, so research extension or education, are eligible for $650,000 for a project that lasts 3 to 5 years. But for integrated projects, the budget request is $1 million for a project that spans between 3 or 5 years. The project types, research, extension, and integrated projects are acceptable in this program. The grant types are similar to those that have been mentioned previously. So the standard, conference, and all of the phase grant types. One of the primary differences between this program and those that have been mentioned previously is the application deadline. So for most of the animal programs, they have a deadline of May 6th at 5 p.m. But for this program, the deadline is July 22nd at 5 p.m. And we have two contacts, myself and also Amr Fayed, who is a national program leader in plant protection. So if you have questions about program fit, you can feel free to contact me and also include Amr, because he and I work through those questions together. Next slide, please. The funding priorities for the tactical science program are the detection and diagnostics of transboundary and emerging pests and diseases of production systems, but also emerging, re-emerging, and invasive diseases, insects, and weeds associated with plant production. So this is where it bridges into the cross-cutting. So it includes both animal and plant-focused projects. And the other focus is rapid response to and recovery from pests and diseases that pose large-scale biosecurity threats to both plant and animal production, especially including existing and imminent threats to agricultural production systems. That's all for me.
Sorry, good afternoon. My name is Andres Sibus. I am a national program leader in the Division of Animal Systems. I'm new to NIFA. I have been here for a month, and I will be describing the next two cross-cutting programs. And so the first one is ideas or interdisciplinary engagement in animal systems. The program code is A1261, and this program funds 3 to 5 year projects for up to $1 million, including indirect costs. And here we are looking for integrated projects. So these are projects that include research and extension or education. And the grant types are standard, conference, and phase grants. And the different types of phase grants are listed there. The application deadline is July 15th at 5 p.m. Eastern Time. And the contacts for this program are Dr. Steve Smith, Dr. Ganesh Bora, and myself, and our email addresses are listed there. Next slide, please. Perfect. So IDEAS seeks to bridge traditional disciplinary divides to address complex issues, in animal agriculture. And it has three broad areas of emphasis. The first is precision animal management. The second is environmental synergies of animal production. And the third is societal aspects of animal welfare. And we would encourage you to check out the RFA for more details on this program, and to contact any of the contacts listed here if you have additional questions. Next slide, please. Okay, the second cross-cutting program that I will try to describe briefly is CARE, or Critical Agricultural Research and Extension Program. In this case, the program area priority code is A1701, and this program funds 1 to 3 year projects for up to $300,000, including indirect costs, of course. And again, just as with IDEAS, the projects here are integrated. In this case, they include research and extension, and the grant types are standard and phase. And within phase, strengthening standard and new investigator grants. The application deadline for CARE is June 17th, again at 5 p.m. And the contacts for this program are Dr. Jim Dobrowolski, Dr. Vijay Nandula, and myself. And our email addresses are shown on this slide. Next slide, please. Thank you. Okay, so just a brief description of care. Again, all of this information is on the RFA, but this program addresses critical challenges and opportunities that research and extension together can address to improve our nation's agricultural and food systems. And the projects here should include integrated activities that involve both rigorous research and effective extension, involvement of stakeholders to develop and rapidly apply new knowledge or practices is important. The focus of this program is to develop knowledge that is ready to apply, ready to be used by the end users. And of course, the projects must contribute to improved well-being of the people, communities, plants, and animals involved in and affected by agriculture and food production systems. Again, as with the previous program, we'd encourage you to check out the RFA and contact us, any of the contacts there if you have any questions. Thank you.
Hello, I'm Ann Stapleton. I'm a National Program Leader at NIFA, and I will give you a brief description of a program called Data Science for Food and Agricultural Systems, DSFAS. This is Program Area Priority Code A1541. This is a change in name and emphasis from a previous program with the same code that was called FACT. Next section, thank you. So there are two types, general types and some special focus areas for our budgets here. 650,000 for standard grants, and for coordination innovation networks, which is a special type, the amount is 1 million per project for up to 5 years. Next. So these are research projects or integrated projects only. Next. And the grant types, as before, include the standard, conference, phase, and all the phase types. Please look at the RFA for the details on coordination networks, because these have special requirements. They're only standard and phase, and they have the higher budget limit and a few additional emphasis areas that you should check. The application deadline is July 29th. 2021. Please contact myself directly, or preferably use our joint email, nifa.dsfas@usda.gov. There are several national program leaders in various areas, specialty areas that are involved in this cross-cutting program. So the appropriate person will reply to your query if you send it to this email box. Next, okay, I want to talk a little bit about the funding priorities for this. Here, it's a cross-cutting program in that this funds research in data science to enable systems and communities to effectively use data, improve research management, and integrate new technologies and approaches in food and agricultural enterprises. Next. This is a question I'm often asked, so I wanted to emphasize it here. The most competitive proposals in this competition will equally strong in agricultural science and data science. So they have strong research questions in both areas, and they bridge these areas. Next. So I just wanted to put a short introduction to the priorities. These are just the main sections, and there's additional detail in the RFA for each of these. So priority areas include analysis of agricultural data, multi-scale, multi-domain agricultural data, and agricultural applications and human technology data interactions. There are examples and criteria for each of these areas in the RFA. Next. So please do see the RFA and write to us if you have any questions. I also wanted to point out there are a few specialty areas in this RFA. There's special emphasis projects in artificial intelligence and machine learning. And there's also quite a bit of additional detail about the coordinated innovation networks projects, if you're interested in those. Thank you.
So this is Deb Hammernick again, and you just heard a nice detailed explanation of the base program in the animal health production and animal products area, Farm Bill priority, as well as some of the cross-cutting programs. But just in case you didn't see a good fit for your specific project, we wanted to draw your attention to several other AFRI programs that might be relevant to work in animal science, and those are listed on this slide. So within the AFRI Foundational and Applied Sciences RFA, there are other programs that deal with antimicrobial resistance, nanotechnology, engineering, small and medium-sized farms, and then the economic and social implications of food and agricultural technologies. So please do check out those program descriptions if you think that might be a good fit for your project. And then we also wanted to call your attention to the AFRI Education and Workforce Development RFA. And there will be a special webinar entirely on this RFA on tomorrow, Wednesday, March 3rd at 1 o'clock Central Time. So if you haven't registered for that, you might be interested in that program. This RFA, if you're looking for training opportunities for pre-doctoral fellows or postdoctoral fellows, these are two opportunities you might be interested in. And then there's also a research for undergraduates, education and extension undergraduate students. That's more of a training grant opportunity. So again, if you're interested in those types of funding opportunities, check out the RFA and try to attend the webinar tomorrow afternoon. Next slide, please. And then there's also some non-AFRI programs in NIFA that have opportunities for funding animal science work. So there's two programs on organic agriculture research and extension or organic transitions. There's a program on beginning farmers and ranchers development program. And this program just received a considerable amount of money, extra money in the COVID supplemental funding appropriations this year. There's a biotechnology risk assessment grants program, and there's a special research grants program for aquaculture research, as well as the small business innovative research for animal production and protection or for aquaculture. So again, there's many opportunities to fund animal science research outside of the AFRI RFA. Next slide, please. And then finally, this is a page that you'll want to make sure you visit as you start to prepare your application, or if you have additional questions that you might think of after this webinar. So you'll get a copy of, when you get the handouts, you get links to these websites that have frequently asked questions for new investigators, general questions, and then regarding the phase program. Last slide, please. And so we're happy to answer your questions now. If you have additional questions, go ahead and type those into the question and answer section at the bottom of your screen. As you probably saw, everything we talked about today came straight from that AFRI Foundational and Applied Sciences RFA. So you can always go back and look and read that RFA for the details. And then finally, if you have questions about whether or not your project is a fit for a specific program, or you're not sure which program you should submit it to, please do reach out and contact the national program leaders that you heard from today. They'll be happy to visit with you, probably in a virtual meeting or a phone call, and answer your questions. But make sure you get your questions asked before you submit an application. So that's it for our presentation. And I think if we stop sharing slides now, we'll be happy to read some questions to you. And we've got at least a half an hour here for questions. So be sure to type your questions in the box and we'll get those answered.
So the first question is pretty broad, but if an EPSCoR, as an EPSCoR institution, are we eligible for 800,000, or is that only if we're collaborating with another EPSCoR institution?
So I probably ought to answer this one. It's a unique wrinkle that was unexpected when we launched that program. So we do allow what is listed as the partnering institutions to be the lead, but they will then need to partner with one of those other institution types as well.
The next question is again about strengthening standard grants. So Mark, if you want to take this one as well. What is the difference between strengthening standard and strengthening seed grants? As a PI at an HBCU that has never had an AFRI grant, should I apply for a strengthening seed grant or a new investigator award?
So you can, if you apply for a seed grant, then it would be, you would actually be applying as both a strengthening seed grant and a new investigator grant. So you would check new investigator, strengthening, and seed grant, hit all those categories. The difference is the award size and duration. So a new investigator standard grant will have the maximum award size allowed. So typically $650,000 for most of those programs that were discussed. Some may be a little bit larger. And the duration would be 3 to 5 years. The project narrative for this new investigator standard grant is up to 18 pages in length. And it is standard grants of any type are expected to have preliminary data. So that is one of the review criteria that is listed in the RFA. New investigator seed grants or strengthening seed grants have a budget limit of $300,000 in total costs for up to 2 years, for 2 years in duration. They have a 7-page project narrative. So the smaller grant size for a limited scope of work. So only 7 pages. They are intended to build capacity and basically to secure preliminary data for inclusion in a subsequent standard grant application to AFRI. So preliminary data is not required nor expected in a seed grant, whether it's strengthening seed grant or whether it's a new investigator seed grant.
And along those same lines, we have another question saying, can you please explain clearly what the requirements are for new investigators? And the second part is, and the differences among standard grants and those under phase subdivisions.
Steve, you want to handle that one?
You're on
You're
mute.
muted.
I'm sorry, I was reading ahead on some of the newly submitted questions, anticipating those, and I missed this question. So could you repeat the question, please, Sierra?
Yes, of course. So it's, can you please explain clearly what the requirements are for new investigators and the differences among standard grants versus those under phase subdivisions?
Okay, take them in reverse order. The qualifications to be considered for a seed grant on the basis of being strengthening, you have to be submitting from a strengthening eligible institution as defined within the RFA in the phase category. With the separation of the new investigator seed grants, that allows for those to be requested by new investigators who are not from strengthening eligible institutions. So that's the big distinction there. As far as the content and the intention of the seed grant, it's the same in both cases to help an investigator develop preliminary data that would make them more competitive when they come back and submit for a standard award. Now, remind me of the first question there, Sierra. First half of that question?
Eligibility.
Eligibility, okay. The eligibility for a new investigator is that you be and less than 5 years into, well, 5 years, no more than 5 years in your, say, professional track career. So there's a little bit of discretion there, but for the most part, that's once you land, if you're working with a university in a tenure track position, and then there's also an exclusion for extensive publications. So for instance, if you are in a postdoctoral position in general, those don't count toward that 5-year limit. However, if you have sequential postdocs and a rather large publication record, probably will tip you over into losing that eligibility. The other thing that will play into that, if you are in a position where you are allowed to submit for grant applications, and I mean other than seed grants, other than fellowship grants, but the competitive grants that we've talked about today. Once you receive one, you may be in your first year as a new investigator, you will no longer be eligible for subsequent new investigator awards, or new investigator considerations. So less than 5 years in your career track position, having not received a federal grant, and that's from all sources. That's not just NIFA. It can be a NIFA or NIH, where you are the PD, the project director, not a co-PD. Those don't count against you. And that you don't have this long track record of publications that would infer that you are more professionally advanced.
Great. So the next question is, can a survey conducted with scientific standards be considered as preliminary data?
A survey, so I guess that's sort of along the lines of epidemiological kind of work. So yeah, surveys, if it's science, that would be survey science, and social scientists would consider that research. So that would be eligible for preliminary data. Preliminary data is really whatever you can convince the reviewers. I'll come out of hiding. Whatever you can use to convince the reviewers that your project has merit and is likely to be, that your hypothesis or goals have merit and that your project is likely to be successful.
May I contribute to that answer? Also, if part of your research is to develop or promote a new animal product, and a question could come around acceptability by the general public, if you have a survey that shows that would be something that would have acceptance in the public, I think that would be a valid point of preliminary data.
All right, so the next question is, if an 1890 institution is the lead and collaborates with a non-EPSCoR university, does the non-EPSCoR have to get a certain amount of the budget? For instance, does the budget need to be split 650,000 and 150,000?
Simple answer, yes. But in that case, the 1890s as the lead institution gets to keep the lion's share of that. But the RFA basically specifies that 150,000 needs to go to the partner institution. And the logic is we want these partnerships that develop to be substantial and significant.
Usually we get asked that question in the reverse, where you've got a 1864, for instance, partnering with an 1890 and wondering, what must be shared with that institution. And we're really trying hard for those not to be just a straw man. You know, they need to be full active participants, because that's what's going to make these beneficial to the to eligible partners. So to have it asked in reverse, I think is quite interesting, but Mark's answer is spot on.
Okay, the next question is, if a new investigator receives a seed grant, are they still eligible for a new investigator standard grant in the future?
So yes, they are. So the RFA says, or the grant types section in the RFA says, new investigators eligibility is that they cannot have received any federal competitive grant funding except for a pre or postdoctoral fellowship or a seed grant. It doesn't specify what type of seed grant. So we have developed these seed grants so that a person could come out, start a career track position, perhaps quickly get a seed grant, and before their eligibility expired, could then progress to applying for and perhaps securing a new investigator standard grant and really get off to a good start.
What about 2 seed grants, Mark?
So you cannot have 2 new investigator seed grants, but if you are at a strengthening eligible institution, you are eligible to have a second strengthening seed grant.
And can you expand quickly on the difference between a new investigator seed grant and a strengthening seed grant?
Sure. The simple answer is eligibility. If you are at a strengthening eligible institution, minority serving institution, or in an EPSCoR state, or small to mid-size institution that is not among the most successful, then you are eligible to, if you reside in one of those institutions, you're eligible for the strengthening award and a strengthening seat. Right? If you are not in one of those institutions, you may still, under these new rules, apply for a new investigator seed grant, and that would be the distinction between the two.
And the additional point would be that you do not have to be a new investigator to apply for a strengthening seed grant. You do have to be a new investigator to apply for a new investigator seed grant.
But please do indicate, we'll catch it anyway, but let us know if you are a new investigator from a strengthening eligible institution applying for a seed grant, because that will, we give those extra consideration.
Okay, so the next question is, do you need to wait until the 2-year period of a new investigator seed grant to be over before applying for a standard new investigator grant?
No,
shaking her head, but
there's no restrictions in the RFA that state that, so you do not need to wait.
And can you define NIFA criteria of extensive publication record for new investigators?
I'll let Mark touch that.
Okay, so the short answer is no. This is a kind of a gray area. That language is pulled directly from the Farm Bill, and so it is really a judgment call. I can say that in 20 years, we've only excluded, I've only ever suggested that we exclude 1 due to extensive publication record, primarily because they were in postdoctoral positions for 12 years before gaining a faculty position. And so they had accrued. That wasn't really the intent of the new investigator grants. It's really early career investigator. I'll just add that if you have questions, you can send us your publication list or your CV, and we can give you a determination. If you mark new investigator and we determine you're not a new investigator, we're still going to evaluate your application as a standard grant. If you applied for a seed grant and you weren't eligible, it would still be evaluated as a standard grant. But we don't kick you out because you are not eligible and inadvertently mark the wrong box. But again, as Deb said, you can reach out to us and we'll give you a determination.
Okay, so this one is a little bit similar to an earlier question, but I am a new investigator in an EPSCoR state. Can I apply for one new investigator seed and another seed based on the EPSCoR?
Yes.
Yes, but not in the same year. You're only allowed to submit one seed grant in any given year to all AFRI programs. So you could apply for a seed grant either as a new investigator or a strengthening seed grant. And then the next year you could apply again to the same program or a different program for another seed grant of the other type.
Since preliminary data are not expected, but also are not forbidden in seed grants, would a seed grant proposal with preliminary data have advantages over another with no preliminary data during review?
Okay, I see Steve smiling and I can't tell whether he's going to answer it. So it depends on the volume of preliminary data. I really think it does, because with too much preliminary data, you run the risk of the reviewer saying, there's an awful lot of preliminary data in here. It seems like this should have been submitted as a standard grant. So if you have a little preliminary data, I don't think there's a lot of harm in it. If you have enough preliminary data to support a standard grant application, that's the route you should go, because you have that information and you can go for the larger grant.
Next question is, are collaborations across institutions encouraged or discouraged in new investigator seed grants?
Steve will answer that.
Yeah, where it's pivotal to the integrity of the proposal, I think that's valuable. It adds, but considering the scale and scope of a seed grant, they're $300,000 and then indirect costs come out of it. That's not a lot of money to share a lot of different ways. So if there is a real valuable reason to be collaborating, to have a multi-institutional application, they're certainly not discouraged. It could strengthen the application if it's done well. But do be mindful as you make these decisions of the scale and the scope and the purpose of the seed grant, which is to help you develop the strength that you need to the capacity that you need to be more competitive in a standard application. So if you're using it to bring in a piece that you can't handle yourself, by all means do. If you're using it to establish some of those collaborations that you may need as you go into your standard application, I think that's valuable as well, as long as the funds and the scope of the project will allow it. But it's certainly not required and not an essential piece.
So additionally, my standard response to that type of question, and whether it's collaborating institutions or co-PDs or collaborators, is the project should drive that sort of effort. You need to do what makes the most sense for the project, and you should never add co-PDs or collaborators or collaborating institutions just for the purpose of window dressing, or because you think it will be perceived better by the reviewers, because usually it will not.
This next question, Tim or Kathy, if you guys want to tackle it. I'm a new investigator. I applied for USDA NIFA standard grant in diseases of agricultural animals last year, but it was not funded. I am planning to resubmit this year. Can I submit a seed grant along with this?
So you are allowed to do that. I don't believe you're allowed to submit 2 grants on the same topic, but a standard grant and a seed grant on different topics would not be disallowed as the program is currently set up. That would be my advice. Although typically you may have better, the panelist is going to see both of those submissions, so you may have better luck putting all of your effort into putting together one really good proposal, but it really depends on the workload and what the 2 projects are. That would be my guidance. Mark can chime in if he has other things he'd like to throw in.
Perfect answer.
At the moment, that was the last question. And Terry has been doing a great job of answering some and typing some of those answers to your questions in the chat box. So make sure to refer to those as well.
Hey, are there any other questions from the participants? Looks like we've lost a few people over time here, so well, looks like there might have been one other question just pop up here.
Yep, one question did, and it says, in my seed grant last year, the reviewers didn't like much of the preliminary data. If I remove it, will it cause some harm or be a disadvantage to that proposal?
What do you think, Deb?
Well, since we don't know what program he submitted to, I was just going to answer. I would encourage you to contact the national program leader for that program and have a conversation with them. They can often help you understand the reviewer's comments and maybe help you understand what was wrong with or what the reviewer didn't like about your preliminary data. But that's a conversation that's probably best held with the specific program officer.
So I would contribute and say that if the preliminary data does not really increase the believability or the credentials of your project, then it's really not healthy. Now, sometimes you might have the right type of data, but it's just not strong enough. Maybe you had a small sample size, or the conditions where you ran that are not similar to the ones that you're proposing to do. So I think in that case, the preliminary data is not really helping you. So it's, as Deb said, talk to the national program leader and they'll maybe be able to walk over it with you.
And sometimes just being able to show that you can conduct a technique successfully in your lab is enough, right? If it's a new assay or a new technique that you've not published on before, it just, a little bit of data showing that you can generate data with this new technique is helpful. So again, there's a lot of ifs here. There's a lot of programs specific components here. So I think the best response would be to ask the program officer. Looks like we have a couple more questions. So how long is
I was just going to chime in and say that I thought those were great answers, but there's lots of reasons that they might not like the data. Some of it is just how it's presented. It may not be that they don't like the data, they just don't understand it. It may not be presented well. The font and the figures may be too small to read, or gels may not, be convincing because they're poor quality images. So there's, as Deb said and Frank said, there's lots of reasons for criticism of the preliminary data. And I think I agree that you need to have that discussion directly with the program leader that oversaw the program and probably has notes on that discussion.
All right, so the next question, how long is the review process? So if Kathy maybe wants to take
the
a stab.
review process, do you mean if you mean from the time of submission to the time that funding notification decision or funding decision is made, then that generally runs from about 4 to 6 months. So that's generally the time span. I will say that in AFRI and generally across NIFA, we try to adhere to a standard of within 30 days after the submission deadline, notifying you that your proposal has been brought into a program and will be evaluated in a specific program area priority. And that email, that should be one of the later email notifications you get, and you'll get that directly from the NPLs overseeing the program. And in that email, one of the things they should be providing is the timeline for returning a funding decision to you. They should give you an approximate date that you should expect to see an email that says congratulations, or that says we hope you will try again next year.
This next question can be for either Steve or Andres. It's about the IDEAS program.
Okay.
How critical is it that the PD of an IDEAS proposal has been previously funded as a PD on a seed grant or standard proposal?
Okay. It is certainly not a requirement. So this is a standard award, much like others, though it is larger in scope, and these are integrated proposals. So the bigger challenge I think that you'll run into if you are in fact a new investigator is just having the network that could pull together an integrated proposal and really make it a compelling and successful application. But I don't mean that to discourage you, because it can be done. These are not cap grants, so that some of those might be out of reach for the experience level of a new investigator. But I would encourage you to take a run at it if your only concern is that you have not been previously funded.
The next question is, can graduates...
Excuse me, Sierra, I'll just
Yeah.
pipe in and add. I was going to let Andres see if he wanted to add something, but I will say that over the last 2 years, the first 2 years of the program, 3 of the 11 grants in IDEAS have been to new investigators.
Mm-hmm.
I'm not aware that any of them had seed grants, although they may have had seed grants, but not from the IDEAS program.
I think, Mark, you and Steve covered it, so I have nothing to add.
Perfect. All right. So the next question is, can graduate students be listed on grants as a co-PD? If they are incoming students, how should this be done?
You're muted, Mark.
So we don't have any prohibition against that, but a co-project director has to be able to take over the grant. If for some reason the project director can't continue on as the lead project director. So in that case, I think that excludes a graduate student from being a co-project director. I think they can be listed as in one of the other categories if you wanted to, collaborator, co-investigator. But that also assumes that they're contributing to the project in a meaningful way, including preparing the grant application. If you don't have their name yet, a lot of people just put to be determined or to be, what's the other acronym there, Steve, that they use for that? To be selected for a future graduate student. If they're at another institution, you can just list them at their previous institution, and that'll be changed when they arrive. If it's really important to list them as collaborators or co-investigators on the grant application.
And the last question I see at the moment is, would you expect too many proposals in new investigator seed grants this year due to its low criteria? And the second question is, what is the average number of new investigator standard grants and success rates in the past? Knowing this ranges across programs, but.
Well, since Mark's not speaking up, I'll jump in and he can fill in what gaps I leave. But in general, we don't collectively track new investigator success. However, our programs within animal systems typically range in the mid to upper 20s lately in success. So 25, 27, 28% is pretty typical. Occasionally we'll hit 30% and if funding is good for, and that's for all standard awards. Within those though, keep in mind that new investigators can be placed by the panel above what we call the funding line, and their new investigator status really isn't a consideration in making that award. But then they get a second bite at the apple if they happen to fall in a high ranking category, but below the funding line, they get special consideration for selection if they're in that situation, they can get a special consideration, and we can reach down below the funding line and make an award to a new investigator there. So their success rates, I don't think are appreciably different than the average investigator, the average contributor or the submitter, probably just a smidgen above average if all things are considered, and all new investigators are counted even if they are above the funding line, which the way we track them, I don't think that those are captured. Mark, your thoughts?
So yeah, we've done an analysis across the entire AFRI program for the past 2 years since we put some additional money into new investigator grants. And across AFRI, all 6 program areas, including animal health and production and animal products, and the cross-cutting programs, we are running right at about, if I remember the data correctly, at about 25% success rate. And it's just slightly across some, it's just all 6, 7 program areas. It's success rates for new investigators is just slightly above that for standard grants. So we have made significant progress in funding additional new investigators in 2019 and 2020. So as far as do we expect more seed grants this year? Definitely, for a number of reasons. One is because the budget limit is higher. Two is we've opened it up to new investigators. The third reason is we've given you a really long time. The RFA was released July 17th, so a lot of people are seeing the RFA and preparing applications. However, we will be using next year, we will have 1.5 years worth of appropriations for our programs. And then the following year, we will also have 1.5 years worth of funding. So 50% more funding per program area priority on average, approximately, is the plan right now. Things could change. We do have a new administration, and they may set priorities and want to do things differently. And, you know, that is what happened back in 2010, when our success rates went from about 18 to 20% down to about 6 or 7% for all standard grants.
One more question here is, would the review panel consider impacts of COVID on preliminary data generation?
I'll take a risk at that answer. I think the answer there, unfortunately, is no, because I can see a lot of people getting creative with the data that they would have generated if they had the opportunity. So I'm not trying to be sarcastic here, but I think that's really going to be a challenge for a lot of people that are going to send proposals and miss the opportunity to generate the data. Maybe one of my colleagues has a different opinion on it.
Yeah, it's hard to, this is completely new ground, so I think it's really challenging to answer that question. Sometimes you can predict what reviewers will say, and sometimes you can't. I will say that just about everybody is in the same boat, right? So it's not like COVID was just impacting one state or one institution or one part of the country. It impacted all across the US fairly equally. So everybody will be impacted by that. And so I think that will sort of even the playing field a bit.
So far, that was the last question that we had.
Okay, I think we'll wrap this up. We've almost been here for an hour and a half, which was our allotted time. So thanks again for attending the webinar, for your excellent questions, your thought-provoking questions, and thanks to the panel, the national program leaders and the program specialists that contributed thoughtful answers and provided an overview of their program. So again, we'll just remind you, we will post a recording on the website if you need to go back and listen to anything or refer a colleague to the recording. We'll share copies of the slides with everybody in the next couple of days, probably next week. And those will have some of the links to the frequently asked questions. Be sure to check those out. Many of the questions that were asked today, I know, are on those FAQ lists. So if you didn't quite understand the answer, you can go back and read that again. To always, you know, remember to feel free to contact your national program leader or your program contacts. So I see there is one more question. I don't know if we want to try. Oh, it just says thank you. So yep, thanks everybody and have a great afternoon.
Thank you, Deb.